So amino acids are subunits of proteins, right? Many people are familiar with proteins, but amino acids build up muscles. They build up glucose and ketone bodies if we don't have enough when we need energy, and as well as kind of the one I like the most. They're neurotransmitter precursors. So, fun fact: something like dopamine is actually only 2 steps away from tyrosine. Right, tyrosine, you're gonna add a hydroxyl group and then cut off that carboxyl group, and you literally have dopamine. And that's very similar in most neurotransmitters, actually. So, the issue that we have with proteins, nitrogen. Nitrogen is very dangerous in our body, right? It builds up as ammonia, and it can cause many different issues. So, like according to Amino Acid Metabolism article, high ammonia levels can cause brain swelling, confusion or disorientation, tremors, and severe cases, seizure or coma. So, clearly, we want to make sure that our body's in balance. So, how do we do this? To do this, we're going to want to kick off that ammonia group off of that amino acid, and this process starts with transamination. So transamination transfers the amino group from amino acid to alpha ketoglutarate, 14 glutamate. You're going to hear glutamate a lot because glutamate is our ammonia carrier. And so if you could see on the top right corner, we have our top right image, and the left we have the amino acid. It's going to go in contact with alpha ketoglutarate, and we're going to get our keto acid. So keto acid is just our acid side chain minus our ammonia. And so the thing that helps me understand where they go in the cycles is looking at the structure of the amino acid, and then you literally just rip off the ammonia, or the NH2, not ammonia, pardon, and literally replace it with a carboxyl group. So on the bottom, that's a great example. See, you have glutamate. Literally just rip off the NH2 and you get alpha ketoglutarate, and that's part of our cycle in the TCA cycle, or it's a functional part in our TCA cycle. So you can say, oh, hey, that's where it might enter. So oxidative deamination. So the important step to understand is that our first part, transamination, was occurring in the cytosol, and deamination is occurring in the mitochondria. So oxidative deamination happens when an enzyme called glutamate dehydrogenase removes the amino acid group as a free ammonia. That free ammonia group then leaves the matrix of the mitochondria, and we're going to go enter the urea cycle. All right. So the urea cycle. So on the bottom right, you can see the urea cycle. So the urea cycle really is a waste cycle. We're trying to help eliminate the ammonia out of our body. So you can see that we start off in the top left. We're going to have CO2. It's going to combine with NH3, and we're going to get carbamoyl phosphate. That carbamoyl phosphate is going to have ornithine carbonyl transferase, sorry if I said the wrong, to citrulline. Citrulline is then we're going to go on arganosuccinate synthase, and we're going to add L-aspartate to arganosuccinate. It says L-aspartate. That's just because our bodies use the L version of these proteins, so it's just a little bit, right, understanding, right? The L is not too specific information to know, because our body isn't going to use it if it's an R anyways. So then arganosuccinate, we're going to have arganosuccinate lyase, turn into L-arginine, and right here is where we're going to see, right, we're tracking the ammonia right now. The L-arginine is going to combine with water, and we're going to kick it out with urea. And so then we're also going to get L-ornithine out, which is our original carrier for CO2 and our NH3. That urea then is going to leave via the bloodstream and get kicked out through the kidneys and our urine. Urea cycle, urine. So this is a better representation, as well as you can see, following those groups around. So now that we have the ammonia group off of our amino acids, what can we use that carbon structure for? So in fuel version, we can use carbon skeletons 2 ways. We can use it ketogenic, or we can use it glucogenic. So it doesn't mean they're strictly one, but leucine and lysine are strictly ketogenic. Others are glucogenic, and then others are also mixing in between. So the definition of if it's ketogenic or glucogenic is where it enters in the cycle. So acetyl-CoA is where we're going to get our ketogenic groups, and then everything else we're going to enter through, right? Remember, if you remember alpha ketoglutarate, it's going to enter the TCA cycle, and alpha ketoglutarate, I think it was from glutamate. I'm pretty sure it was from glutamate, but that's just where it enters in the cycle. Let's see. So the pathway depends where the amino acid turns on, and this actually affects up and down regulation, which is the next slide. So upregulation of amino acid catabolism, or the breakdown. If we have high protein intake, where we're going to have a high amino acid, so it means we can break it down, kick out the amino group, as well as use the rest of the carbon structure. If we're fasting, so we have low sugar, we're going to use those amino acids in our bloodstream or whatever to go create sugars. We have high glucagon and high cortisol. Those are kind of two similar things. High hormones, which means they're low blood sugar. Or if we have high amino acid levels, which means we have excess, we're just going to go use it. We have to break it down if we want to add it back to storage too. Some down regulations of amino acid catabolism. So if you have high insulin, that's going to mean that we're going to have a lot of high blood sugar. We don't really need those amino acids to get broken down for us. High ATP energy levels, that's also going to cause us, right? We don't need sugars again. It goes back on our body's needs. Liver failure, that's just kind of an understanding of where amino acid catabolism happens, right? Deamination happens in the liver, right? And then low amino acid availability is like we're saying, we've been using it for a lot of different things. So cellular damage can actually cause low amino acid availability. So that's kind of the basis of amino acid